Okay, hello everyone. Welcome to the second talk of this session. The speaker is Professor Chenglong Bao from YMSC Tsinghua and Pisa. He is an expert in computational cryo-EM and optimization methods. And today, Professor Bao will present recent computational approaches in cryogenic electron microscopy toward formalization in data-driven cycle. So please join me in welcoming Professor.
Uh, okay. Thanks for the introduction, and I'm happy to present our work in this big conference, and I hope to take this opportunity to introduce our recent works on CRY-EM by using some mathematical approaches and AI methods. Okay. This is the outline of my talk. I will present 3 works and some extensions or ongoing works. So I hope I can explain why I needed to focus on these problems. Nowadays, I think a hot topic is AI for science. But as an applied mathematician, we need to choose some scientific problems that have some impact on science area. I'm working on image processing. So in this area, I think if we take some micro structure, this should have some science discoveries behind these microstructures. So as you can see here, from this 6 scale imaging techniques, all these NMR spectral and X-ray cross-scrapy or light super resolution microscopy. All these methods has been awarded the Nobel Prize. Also, the CRY-EM as an electron microscopy imaging method is also awarded the Nobel Prize in 2017. And this year, the electron microscopy has been awarded the Shaw Prize recently. So as you can see here, the electron microscopy, the image scale is around 10 nanometers to 10 milliamp. And the resolution is near the atomic resolution, like the 1 astron. Okay. Okay, now I hope I can briefly introduce the CRY-EM. As we know that proteins have 4 structures, and the CRY-EM is trying to determine the 3rd structure or the 4th structure of proteins. However, when we want to see clearly the proteins, we hope to have some enough resolution such that we can think the secondary structures, like the alpha helix or beta sheet. If we want to resolve these secondary structures, we need some resolution requirements, such as for different structures, we need different resolutions. And you can see here, the resolution is lower, means this number is lower, means that the resolution is higher. And also sometimes we try to find the side chains, which is important for the function of the proteins. So we need some higher resolutions. And also, as you can see from this picture, we can see if we want to see more details and the resolution is higher and higher. However, the challenge lies in this perspective, which means that our sample, like the biological sample, can be 10 or 10 to 5 times larger than our resolution requirements. So this is we face a big computational challenge in CRY-EM. Okay. Mathematically, I think I hope we can introduce some mathematical model for this imaging modality. As you can see here, these small molecules, which were frozen in this part, and we have some electrons go through along the z directions. And here we have a detector. This detector can be see the 2D projections of these molecules. And also this is our forward model, which means that we assume that the the three-dimensional structure of this protein is X, and we have do some rotation, or this rotation, this Ri, which were unknown, and here we compared to each molecule, this Ri is are different, and we have some projections along Z axis. This is a PZ, and in this detector we have some the contrast transfer functions, and this is HI, and plus some noise. We have our observation. Using these equations, we hope we can reconstruct the high resolution structures of these proteins, which I hope to reconstruct this X. In comparison to CT, I think this is a common application in medical image, and also it has similar formulations. Like we have some this linear model along the projection along certain angle, and however, in CT we know this projection angle, and we know these forward models clearly, and in cryo-EM, our this parameters like this pose parameter or these parameters in CTF were unknown. So if I hope to solve this equation, this is a non-convex problems, and we need to estimate the parameters first and solve this equation, which means that we have some alternative method to solving this equation. Okay, and I briefly summarize the computational challenge in cryo-EM. So first of all, I think the main difficult part is extremely high noise level, which means that the signal to noise ratio is negative. This means that the noise strength is larger than signal strength. And as we know that if in this extremely high noise region, if I hope to solve the true three-dimensional structure, it becomes very difficult problem. And also, these proteins, these projections is provided by the biological experiments, and we cannot control the pose of these proteins. So, which means that we do not know the orientation of these 2D projections, and we hope to estimate these parameters in this high noise data. So this becomes another difficult, and also in experiments we always have highly imbalanced observations. And also the third difficult is data heterogeneity, which means that in this model the X is fixed. However, this proteins is dynamic, and this X is can be changed with these projections. So, which means that we need to solve these heterogeneities in this model. It contains two heterogeneity, which is one is a conformation heterogeneity, which means that each conformation, each xj has some deformation. And also we have some composition heterogeneity, means that we have two proteins binding together. So in this case, this xj and xj plus 1, you do not directly have some deformation difference. We have add some new proteins. And the computation, we face to some massive data set and high-dimensional, three-dimensional reconstruction. Always we need to use some computation in our clusters or with intensive GPU resources. Okay, I show some examples. This 10 protein data set, and we can see that these projections are very noisy. If we want to directly figure out the features of this data from these observations, it becomes a very challenging task. Okay, and this part is, as I mentioned before, this image processing in cryo-EM is a very long process, and this is a highly dependent on the expertise in wet labs. And this figures is provided by the uh, Amazon AWS. This provided some computational resources for this image processing, and we can see this part micrograph movies are our raw data, and we need to follow all these steps, and we can output a final map of these three-dimensional structures. And all this yellow part, and this can be accelerated by the GPU, and we can accelerate our process. Okay. Okay, and then I return to my talk. And as we mentioned before, our talk contains these four components. The first part is that how to estimate the quality of the experimental data in CryoEM. As we see, these data are very noisy. How to estimate this data? And the second part is the data generation, which means that always we try to analyze our experimental data, but this experimental data is highly imbalanced. We hope to using some generative methods such that we can align this experimental data and simulated data such that we can facilitate our analysis. And also, we hope to use some image processing method if our observation is very small, and we hope to using some method to improve the resolution of this three density map. And finally, I hope we still have powerful tools like the AlphaFold, like any other these models. We can combine this with the CryoEM data such that we can output some more accurate estimation using some these CryoEM observations. Okay, so let's go to the first part, the data evaluation. We present some work called the CryoSieve, which is to identify the finest subset to the theoretical limit. Okay, first of all, let's give you some definition of the final stack in CryoEM. Final stack means that we use these 2D projections for the final round reconstruction. As we see that we collect some raw data, we need to select these particles, or which is equivalent to the 2D projections iteratively. And after many rounds of this selection, we can formalize some final stack. And this final stack has parameter estimation and have this particle information. We use these particles to reconstruct three-dimensional structure. And this shows you some typical process for this protein PFCRT. And in original data, you can see that we have over 1 million particles. And in the final round of this reconstruction, we only have 17,000 particles. So which means that we face multiple rounds of this particle selection, and this formalize the final stack of this CryoEM data, and we can using this 17,000 particles to reconstruct a high resolution map. And however, we have one motivation is that, which means that is the final stack still contains some redundancy, which means that we can we reduce this number to a small number. Okay, this is a very difficult problem, as we can see that this is some original data in the final stack, and it's hard to say which one is better than the others. I think we need to formalize some mathematical formula such that we can determine the quality of these particles. We give some ranking algorithm. Okay, why we need to take this part? This is because we still have some theoretical implication for which means that if I have several 2D projections, if I want to reconstruct some three-dimensional structure to certain kind of the resolution, what's the least particles we need for the reconstruction? So this is given by Rosenthal and Henderson. I think this is 2003. No, this is a typo. So which means that here the D is the resolution we required, and here this is some parameters determined by our electron microscopy. And the n particles means that the least number of the particles needed for this reconstruction. And you can see that in these proteins, we found that the theoretical prediction is around the 4,000. However, in experiments, we need like the 17,000. So we have some big gap between our theoretical prediction and our experimental observations. And later, I will show that this is not some unique case, or this is not a cherry pick. All these 8 data set, and as far as we can find from the EM-PAL data set, we can have this for these high resolution reconstructions. The third column is the theoretical prediction, and the last column is the experiment data set. And we can see that huge gap always exists for all these data sets. And so can we reduce this gap with some new evaluation indicator? Okay, so this, I began to say some design of the Cry-sieve. It's very, very simple idea. So I call the B is our observed experimental data, like the 2D objections. And we have some X is our reconstructed map, but this is not accurate. This is just an estimation. And the A tilde consists of the estimated physical model. This means that we combine all these pose estimator or some CTF estimator together, and also this is some estimated model. And we give some G. G means that this is equal to the HB, the norm square of the HB, which means that this is a high frequency part of this observation, minus the second part is the reprojection error of the high frequency part of the reprojection error. So which means that if this G is larger, means that the second part here is near to 0. So which means that this reprojection error is small, this high frequency part. So this gives some intuitive idea. So higher G indicates some better particle quality. And if this is a validate estimating or statistical tool for these particles, and we can use these particles to rank these particles, and we can discard some particles if this score is low. Okay, so I give some certain mathematical justification why this intuitive idea is correct. So this means that if I have two particles, one is good, one is bad. And we hope that this is the basic model of these two particles. This is Bi is equal to the AIXI plus epsilon I. If I assume that we have some certain noise, of course, we can extend to this noise distribution to some sub-Gaussian families. And what we hope to prove is that we need to show that this score is bigger than the second score. The probability is high. So which means that if this probability is high, so we have a bigger chance to identify some good particles than the bad particles. So we can derive some formula for this using some calculation for this score, and we can see that this is determined by the error functions of this part, and this part clearly it contains two errors. One error is the deviation of this density map, which means that our estimate the density map has some errors of our ground truth. And also we have some errors in our parameter estimations. This is also the errors. Okay, so this is, I give you the simulation data for these 4 different errors with different SNR strengths. So if the SNR is 001 or 001, So we can see that we have some big high probabilities to identify these errors. Okay. Okay, now let's go back to our workflow to this corrosive. If this score is good or reliable, then we can use this score to discard some particles. So we design this workflow such that we can reduce the number of particles in our final stack. Okay, this part is actually the standard process, like the half set analysis in Cryem. Okay. I directly show the results. As I mentioned before, we have 8 data sets, and these are high resolution reconstruction. And I think the number in practice we can reduce like this PFCRT data set. We can reduce from the 17,000 to 4,500. And we compare this with our theoretical predictions, and this is near to our theoretical predictions. So I think this is a surprising finding in this area, because as we know, this final stack has been already selected manually or by any other algorithm extensively. However, we still find that over half of these particles in final stack are still can be discarded. However, we still face another problem. We still have some gaps for several other data sets. But this has some difference between your sample preparation or like the electron microscopy, and because these proteins are published in different years. Okay. Okay, and I think the other results I hope to share with you is that the radiation damage. So this is because our proteins can be easily affected by this electron dose, and this is because if I consider different level of this radiation damage, like this is I think this is this different the dose weights. And we can show that our corrosive score first selected the particles with more severe the radiation damage, and then we can keep all the best or the smallest the electron dose in our data set. So okay. And I think in this work, we also introduce some results such that few these particles can have some better reconstructions. And however, I hope to emphasize that we can have some ongoing work on this part, because this theoretical limit is derived 20 years ago. And nowadays, we have some progress in our equipment, or we still need to refine this theoretical limit such that the assumptions can be weaker. And also, as we know, we identify many particles in final stack are not useful. However, we hope to identify the source of these particles, or why these particles exist in our samples. And this should be provided some guidance for the biological experiment list, such that we can have some guidance for the sample preparations. Okay, as this part is published 2 years ago, and we also provide some code and some video reproductions for this work. Okay. The second part is I hope to give you some example. We can do some data generation in Cryem, such that we can solve some challenge problems in this part. Okay, and as we know, deep learning has been applied in many years. Also this method has been applied in Cryem recent years by many companies or universities or research institutes. And we hope to combine these methods such that in all these uh, phases, different phases. And I think the main part of the current AI-based data analysis using in this framework, first of all, we have some experiment data. We hope to train some AI-based representation. And we can analyze this data in several the so-called the latent space. And then we can find some new phenomena or get some new results. However, and we hope to using this co-optimization framework. This is because we have some experimental data, and we're using some AI-based method to do some data generation. And then we hope we can combine this generate data and experimental data such that we can have some co-data analysis. And this is, I think, the main idea behind this framework is that if I hope to find some new scientific discovery, I believe this should be some real events. And so if this is a real event in our experimental data, you face to a highly imbalanced case. However, I need to emphasize these real events. The best way is I can have some simulator such that we can align this experimental data and our physical model, such that we can push up the probability to find out these real events. Okay. Okay, this is an example in Cryem, which is a so-called preferred orientation problem. Ideally, as I say, this protein is frozen in these ice layers, and ideally, we have many 2D projections of these proteins. And we hope these poles of these proteins can be uniformly distributed. So in that way, we can construct a high-resolution map. However, experimentally, we always face some preferred orientation problem, which means that this protein has some dominant poles. This dominant poles is caused by some biological fun properties of these proteins. This is because, for example, this is a membrane proteins, and this membrane is one way is outside the cell, one way is inside the cell. So this protein must have some physical model to the water or this carbon. So this is the main reason to cause the preferred orientation. So your favorite the proteins for drug discovery or other parts, you always face these problems in experiment. So if we face this problem, this reconstruction, you have a big artifacts in your reconstructions. Okay, ideally, and you can see that our experimentalists try to optimizing the sample preparation such that we have some a good post distribution of these samples. And also we can optimizing some data collection. We can invent some new techniques, such as for this part, we can using some of the tilt techniques, which means that here we have some uniform or dominant distribution of these proteins. However, we can rotate this part such that we have more observation of this observation. However, if I using these new data collection techniques, we face other problems. The first problem is particles are more noise. This is because the total dose of electron is fixed. Otherwise, our samples will be destroyed. And also, if I rotate these samples, this part have some, we face some the thickness of the sample. So the thickness of this sample are not uniform. We need to some more computation or analysis for this part. I think the most important part is the throughput is lower, is about the 1/13 of the classical collection strategy. This is because this part is in we need more time to do some alignment of this data if we want to use this new data collection technique. So as far as I know, many labs cannot directly using this data collection method to give to have good results. Okay, I show you some example. This example is this is one data set from the EMPEL 10097. This part means that I using the tilt technique to collect the data, and you can see that we can achieve a very good resolution. And this part is using some classical method to collect the data. And this is a pose distribution. We have a dominant pose in this direction. And we have some severe preferred orientation artifacts. And our question is that, can we directly design some computational method working on this part such that I can achieve similar resolution to this data set? If I have one, then our data collection method can be 30 times faster than this method. So this is our question. So we directly hope to design some computational method which to working on this data set. Okay, so first of all, I think we need to identify the difficult part or the computational bottleneck of this problem. So, and as we know that this is the pose distribution of this data set. If I know the pose correctly, then we can reconstruct a high resolution map. So this means that our data set still contains enough information such that we can achieve the high resolution reconstruction. However, the difficult part is that if I don't know this pose, I need to use some software to estimate this parameter in our experiments. If I do this part, then I have nearly 30% particles have wrong pose estimation. Then it leads to the preferred orientation artifacts in your final reconstruction. So this means that this highly imbalanced data distribution leads to some severe the pose estimation errors, and then leads to these preferred orientation artifacts in your reconstruction. So our idea is I try to get some simulated data such that I can make this distribution be a more balanced distribution and to achieve some accurate pose estimation. Then I can get a good reconstruction. So this is a basic idea. So as I said before, this using the preferred idea. So I have some input. The input is some rough model. This rough model is not correct. You can derive it from the AlphaFold or other knowledge. And this is a low resolution part. And also we have this pose estimator for this raw data. This raw data is from our experiments. And we can use this physical model to get some projections. This is simulated data. However, the biggest issue lies here. I need to align this projection and this raw data such that I can using some generative model to generate several generated data. And then I combine these two data together such that I can get good estimation of poses for the experimental data. So our idea is very straightforward. I hope to get some the generative model such that we can align these two data distribution become to be a constant. Okay. So mathematically, I think we can formulate it as an inverse problem. This is because, as I call this X is our simulated 2D projections, and Y is our experimental data. And our problem is, in fact, is the so-called the unpaired data learning formulation. So which means that I have a marginal distribution of x and y. I hope to get the conditional distribution. Then I can do some sampling such that I can get the synthetic data. So this is the basic idea, the basic mathematical model for this part. And in general, we know this inverse problems is posed because we hope to reconstruct this conditional distribution is equivalent to reconstruct this joint distribution. And I also have some marginal distribution. I hope to reconstruct the joint distribution. Your observation is clearly insufficient. Otherwise, you can assume that we have some independence of the x and y. For example, if x and y are independent, we know pxy is equal to px times py. Clearly, here we cannot assume this independence. Okay, so we designed some method we call the learning from unpaired data. We propose some this generation graph such that this is the x part only compared to some z, and this part is compared to corresponding to z and zn. If I have this generative graph, then we can design some generative model and inference model. And in this inference model, clearly the red part, the Q part, z giving x and y, we still need some paired data information. This is because we only need this x and y is a paired data, and it influences the common latent space. So this is a general idea. However, we need to relax this part such that we can obtain some trainable loss in our model. Okay, so this is our inference environment condition. So which means that if I have some pair of this training data, I hope to assume that QZ giving X is equal to QZ giving Y, which means that if I have a pair data, no matter this is a noise image or clean image, they share some common representation. This is a basic idea. If I have this inference environment condition, then we can only need the samples from X domain and the samples from Y domain. However, when this inference environment condition holds, we can prove that this condition holds asymptotically if I apply some preprocessor to this X and Y together. So this is, we have some theorem to show it. And nowadays, we can prove that if H has some better multi-scale representation of this data, this condition can have a better rate or any other things. Okay. Anyway, this is a quantitative evaluation of this method. We cannot directly evaluate on the Cry-EM data set because we don't know the ground truth. But we evaluate on this the smartphone imaging. This is a classical test in computer vision, I think. So we use the FID, AKLD, this indicator to show that our model can learn this complex noise distribution very well. Okay. Okay, let's go back to our Cry-EM. Okay, this is a generation from our model. You can see that the first row is the experimental data, and the second row is our generated data. And after generation, this is a post-distribution. You can see that we make this distribution more balanced if this generative data is reliable. Okay. However, as we can see in this picture, we cannot get more information because the noise is very, very high. And after 2D classification, the first row is still the experimental data, and the second row is our generative data. And you can see many different orientations in this 2D projection in our after 2D classification. So this means that clearly we generate some more more projections from different poses with different poses. Okay. And here I hope I can show that this generation data combined with this experimental data, we can reduce the error of the post estimation. So this is an example. So this figure gives the reprojection error in the classical data set. And this gives the error of our co-data set. So this means that after I generated this data, we can significantly reduce the error of the post estimations. If I achieve a better post estimation, we can make this density map from this part to this part. So we have a big improvements in this simulation results, right? Okay. So refer to our previous example as I showed before. The left part is I use some experimental technique such that we can achieve a good resolution. And in this data set, we can also use in our method to achieve a very similar resolution, right? This is very comparable. We can have some big improvements from this part to this part. Okay. Okay, now we can apply this to many different biological labs. And these labs which means I sell this method to these wet labs. I just say we want to recycle your data set. We try to apply this to the called garbage data set in these labs. So this is one from the Yanling's lab. So this is original data distribution, and using their method only achieve very small resolution. And using our method, we can have a big improvements. Okay. Also, we apply this method to the data set from the Huqi's lab. This guy is from the Westlake University. And this is their original reconstruction. You have some 8 astrom resolution. And after our analysis, we find some new density between these two proteins can be binded together. So this should be some new discovery or any other we need to be verified with this person. Okay, anyway, in our paper, we only want to show we have a better resolution reconstruction. Okay, now I think this method seems good. However, we hope to discuss some risk behind this method. This is because we're using some generative model, that this generative model can introduce some hallucinations. We hope we cannot directly fully solve this issue, but we hope we can discuss this issue such that we can have some realization or we can have some progress on this direction if I want to claim we have some new protein structures. So this the original issue comes from the particle picking. As I said, we need to pick some the signals from the raw data, and these green cycles means our signals, and this green cycle is introduced from the alignment from a template, which means that it calculates the cross correlation and using this cross correlation to select these signals. So this means that your selected this data can introduce some incorrect information from your template, especially for high frequency part. So this is a big risk. Okay, give you a experiment, which means that if I have R, this is a template. I have multiple Gaussian distributions, and I'm using some alignment for the template and this Gaussian noise. And then we average this aligned image. We can see that this average image that contains some high frequency information from this template. So this means that if I can directly restore some high frequency information from our template. This directly shows some risk in your final round reconstruction. Okay, this is also have some debate in this community, which is called the Einstein from noise. In 2013, we have published a structure of this HIV, and we have 3 discussion papers related to the reliability of the reconstruction. You can directly see the results, the conclusion from this discussion. This is finding the structure from the random noise. Or we need to pay more attention for this model reliability. And they also have some reply to this discussion to show that we does reconstruction from these 2 signals. So this is an unsolved issues in this community. And also nowadays, if we hope to apply these generative models or foundation models to this part, we still need to validate or to certain kind this reliability of our reconstruction. Okay, in our paper, we cannot fully solve this part, but we do some experiments. The first experiment is from the data perspective. As we know that the pure Gaussian noise, its average is 0. However, in our experiments, we have a severe signal-dependent noise. So this is the average of the pure noise or from our generative noise. This is an indirect proof of this noise, the generative noise. And the second part is from the model perspective. From the model perspective means that I have some initial template. I hope the unique high frequency information in our template disappears in our final reconstruction. And also the unique high frequency part information in our final reconstruction does not contain in our template. So this gives you some result on these 2 perspectives. Okay. Okay, this is also published in this year, and also we have some method which is published in computer vision conference or journals or anyway. We publish all this data set, and we give tutorials and results to you can reproduce these results. Okay. Okay, I think I do not have too much time. I will try to accelerate this talk by briefly introduce the post-processing, the prediction high-frequency resolution of real conformations. This comes from this intuition. As I said, that the proteins is dynamic, and a different conformation that's corresponding to the different functions. And as we see, this is the SARS-CoV-19, and we have the 3 RBD domains in these proteins, and each RBD domain up is related to the possibility of infection of this SARS-CoV-2. Okay, so this means that I hope in our samples I can resolve all these 4 conformations together. Okay, so this gives our mathematical formula. This is a heterogeneous reconstruction, which means that our x here becomes to xj, and then xj follows from some distribution in your energy landscape. Okay. Okay, and this is, you can see that this is in a real data set. You can see that this data set that contains 5 different conformations, and each conformation has different resolution. This means that if this conformation has many particles, we can have a good reconstruction. If this conformation does not have enough observations, you have bad reconstruction. So we call that this conformation is a real conformation, which means that this is not easy to observe, and also this real conformation is corresponding to the high energy states. So it's not easy to observe. The total number of observation is low. So this means that the reconstruction resolution is relatively low. And our idea is I can use some method to increase resolution for this part. Okay, mathematically, you can see we have this problem. We have some minimal 2D projections. We have a high resolution reconstruction. And also for some real conformation, we have many few particles. We have some low resolution reconstruction. So our idea is we try to find some deformation T acted on this F0 should be a high resolution prediction of this F1. So this is our mathematical problem. Okay, so we provide some of the neural ODE based models, which means that I need to represent this deformation using some velocity field. And this velocity field can be parameterized by a neural network. So this is a representation. And in this classical variation formulation, we need to determine the loss function to compare your prediction and your observation. So here we use the Wasserstein distance. And also I need to impose some regularization to the velocity field such that this trajectory can have some physical constraints, right? Okay. So this is our model. We have some high resolution input, and we do some new deformation, and we obtain some generative map. And this generative map has some loss between our low resolution target map. We need to compare it using the Wasserstein distance, and we can show that this Wasserstein distance should be better than our classical L2 distance in this case. This is, I will explain later. And also we need to add some physical constraints to our velocity field such that this change has some better regularization. Okay. So the first part is I hope to explain why Wasserstein distance is a favorable distance in this part. First of all, we can consider the loss energy landscape. As you can see that this part is a Wasserstein distance, and this part is a classical Euclidean distance, the L2 norm. And we can see that if I want to use a Wasserstein distance, it gives you a more convex loss landscape, and we can have some effective gradient if I do some backpropagation. And also we can prove some result. This result, I think this should be a very interesting result. This is because I need to do some backpropagation. I need to calculate our desired gradient in compared to our data gradient. So which means that I hope to calculate the gradient between our two high resolution density maps. But what I only have one high resolution and one low resolution. This is because our target map is a low resolution. So we can prove that if we use the Wasserstein distance, this gradient error is less than our classical L2 norm based error. So which means that if I do some backpropagation, this gradient is more close to what you want is this gradient. Okay. Okay, and also I need to show some regularization for this velocity field. And in this velocity field, we hope in this proton deformation, we have some local rigid deformation and large non-rigid deformation. This is because for secondary structures, this deformation should be rigid. And in this third structure, or as a whole, this should be a non-rigid deformation. So when we use some neural network representation, we impose some non-rigid deformation. However, we need to add some regularization to this local rigid deformation. Here, we're using this regularization. We can see that locally, we transport from this mesh to this mesh. And this mesh transformation uniquely determine a linear transformation. We can penalize the eigenvalue of the linear transformation, and we know in 2D case this is directly the quasi-conformal mapping. Okay. Okay, I show some results. First, I give you some tests on this simulated data, and we can see this is our initial map, and this is the target map with different resolutions. And we can see this part is our prediction. It still keeps a good resolution. Okay, and also we apply this to some experimental data. This data is given by the Zhuping from the Chinese Academy of Sciences. And this is the initial map, and this is a target map, and this is our generated map. And I think the easiest way you can just see this number. This number is smaller is the better. So we can achieve a very good and also we can apply this method to some data from different labs. And this initial lab map is from some Chinese lab, and this target map is from some UK lab. And we can also have this registration such that we can do a better alignment or prediction. And also we can register from different modality. And this part is from the SPA, and this part is from the ET. This is in vivo reconstruction. Always have some low reconstruction, low resolution reconstruction. And this is our prediction. Okay. Okay, this is to show you why we need to add this regularization to the velocity field. And you can see that if without this regularization, you have some unmeaningful this local strength of this density map. So this is not realistic. So we hope this regularization can help us solve this issue. Okay. Finally, I think this is just a prediction using some image processing method. However, our review still need us to validate using some different method. Here we validate our trajectory, the method from molecular dynamics, and we can think that in this case, our trajectory should be very consistent with our result from the molecular dynamics. Okay, this paper is also published. We also publish our data. Okay, finally, this method is to show how to incorporate the sequence information. We know AlphaFold or any other protein fold model give us a very good results. I hope to show one is from the AlphaFold. These are two experimental data from some labs, and we can see that the AlphaFold cannot have a good prediction for these models directly if this experiment is new, or this experiment we need to solve some more complex proteins. And here we give some results such that we hope to give some tunable AlphaFold such that we can have some more accurate predictions of the structures given by AlphaFold. Okay, I think we hope to upload this preprint, this manuscript recently. Okay, I hope I should summarize my talk now. In this talk, I show you some results on or your data evaluation, data generation, and post-processing. But this, how to directly incorporate this AI-based method to the whole process in cryo-EM reconstruction. This is still have some many more challenges or many more problems need to be solved in future, I think. However, I hope it to discuss some AI for industry versus AI for science. I hope to emphasize what I need in AI for science. The most important part is to validate our understand our discoveries is true. This may be a good the ultimate goal in this case. However, we hope as a prime mathematician, we hope to design some computational tools such that we can do some cross-validation between our computational method, different computational method, and have some statistical guarantees. Okay. Okay, many works go, I think the first part is that we have some mathematical analysis in our general data generation for the unpaired data or semi-paired data. I think this is important to simulate our data such that we can combine it with our experimental data. Okay, I think I stop here. Thank you very much.
Okay, thank you, Professor Bao, for the interesting talk. So, are there any questions?
Because I do lots of interdisciplinary research.
Yeah.
I'm curious about your last point in the conclusion. If
Yeah.
you can explain.
Yeah, thank you for your question. This is in China, when I go to this conference, I will emphasize this part. This is because when we do some interdisciplinary research, we apply our method to do some experimental data. In many cases, this data is published, which means that the experimentalists have achieved some goal using this data. What we do is we reproduce this success using this data set. However, many future case that's not published, but we don't know this case. We do not have these resources. But what I believe is that if I can find some computational method such that we can work in this future data sets with certain factors, this must have some new insights or some new tools for these different research areas, which may encourage the interdisciplinary collaborations across different fields. Because currently, I show this experimental data set to our audience, but this is because what we do is we only do it using our personal connection or personal communication, but we don't have any this data platform such that we can directly working on this without this. Yeah, this. Thank you.
Any other questions? I also have a quick question. You talked about extremely noisy cryo-EM images, and you talked about generative models. So, and I wonder if neural network like diffusion probabilistic models can help remove noise from that's extremely noisy images by simulating how physical processes like diffusion, clean the data. I don't know if it works or.
Yeah, thank you for your question. Yes, I hope I hope to use this picture to answer your question. This is because, as you say, we can apply many denoising methods such that we can amplify our signals. But if you apply any denoising method, you must destroy some high frequency information, because you in certain kind you do some filtering techniques, right? And what's the most effective way is I need to do average. if I have multiple observation or many observation. So this means that if I apply some denoising method to our original data, you cannot directly have a better resolution in your final reconstruction, because your better denoising denoising algorithm can have some effect on many this part, can be affected in this part. And also I also want to mention that the denoising method is useful in certain kind. This means that I don't use the denoise data to do final reconstruction. I can use the denoise data to do the classification or do parameter estimation in this part. But in final round reconstruction, we prefer the original data. Yes. Okay. Thank you.
Any other questions? So if there is no more question, you have a question.
Maybe I missed a part of the talk, so can you explain a little bit more about your criteria for filtering out the bad particles? Filtering out the bad particles, the criteria.
Okay. Okay. So you mean this score,
Yeah,
right?
yeah, exactly.
Okay. This is our observation. It's our collection. So this is a high frequency part of this our observation.
Yes.
And this part is our the reprojection error. And this is a high frequency part of the reprojection error. So which means that your reprojection error is very small. Then it means that this part have better high frequency alignment between the projection and your observed data.
I see.
So this means that this particle is more reliable in your high frequency part.
I see. Okay,
yes.
That makes sense.
Okay.
Yeah, thanks.
Okay.
Any other questions? So if there is no more question, please join me in thanking Professor Bao once.
Thank you.